Now my distinct pleasure to introduce today's moderator, Shui-Ya Wang, PhD candidate at Westlake University in the lab of Professor Guo, and holds a bachelor's degree in biotechnology from Nantan University and a master's degree in biochemistry and molecular biology from Fudan University. Following his postgraduate training, he worked at the bioinformatics as a bioinformatics algorithms engineer in the bioinformatics department at Hangzhou PTM, where he contributed to the development of computational tools for biological data analysis. In 2024, he joined the PhD program in the School of Life Sciences at Westlake University, where he is currently engaged in AI-driven research projects at ProteomEx, with a focus on integrating artificial intelligence and bioinformatics to advance life science research. And with that, I will hand it over to you.
Okay, thanks for Renee's introduction. So it's my pleasure to introduce today's speaker, Dr. Zheng. Dr. Zheng is a research assistant professor in the lab of Professor Tiannan Guo at the Westlake Laboratory of Life Sciences and Biomedicine. He received his PhD from the University of Otago in 2020 and later conducted postdoctoral research at Westlake University, focusing on the development of expansion-based spatial proteomics technologies. So he has received several competitive awards and grants and has published as first or corresponding author in leading journals including Nature Communications, Nature Protocols, Hepatology, and so on. And Dr. Zheng co-developed the first generation expansion spatial proteomics technology, ProteomEx, and lead the development of a second generation method, FAXP. These technologies were recently highlighted in Nature's technology features, reflecting their impact on advancing spatial proteomics. And today, he will share his latest work in this exciting and rapidly evolving field. So please join me in welcoming Dr. Zheng. Yeah.
Okay, so I will start. Hello, everyone. First, I would express my gratitude to Bell Protocol for the invitation and thank my moderator, Mr. Shui-Ya Wang, who is also my collaborator, for the kind introduction. And today, I'm honored to introduce our spatial proteomics workflow, namely the expansion proteomics and its applications. And this talk is mainly about these three parts. Firstly, I will talk a little bit about the background of the mass spectrometry-based proteomics and expansion proteomics. Secondly, I will talk about the expansion proteomics iterations and their applications. Last but not least, I will talk about the spatial proteomics, mainly the expansion proteomics, and how this acting as a foundation for the virtual cells. So for the first part, about proteomics. Over the past decade, the mass spectrometry-based proteomics has become a mature and powerful platform for the biological research, especially the clinical research. And we can now analyze almost all kinds of samples, like the liquid, serum, urine, tear, and also the fresh frozen tissues, FFPE tissues, and even bones and freckles. So kind of basically we can analyze all kinds of samples. And there are quite a few advances in sample preparations and in the liquid chromatography, short as RC, and MS based proteomics, instrumentation, and different types of data acquisition and data analysis. So all these advances have made the large-scale clinical proteomics study feasible and robust. And more recently, like last year, my lab PI has wrote a review in Nature about the most advanced technology happened in the mass spec-based proteomics. And from this part, we can see that now, earlier when I mentioned we can analyze all kinds of samples, and now we can analyze very small amount of samples down to, for example, the single cells. And all these advances also are accelerating the rise of the spatial proteomics, which is the topic today. And all those developments that would have seemed unrealistic just in several years ago. And to note, the spatial proteomics was named the method of year 2024 by the Nature Methods. And making this area as a technological frontier for like single cell analysis, like PTM, meaning the post-translational modifications, the functional part, and also the multimodal data integration. Actually, in this spatial proteomics umbrella, there are mainly two approaches. One, imaging-based, or you can call it affinity-based, like Codex, CySIF, just like the immunofluorescence method. And another one is the RCMS-based method. And among these two methods, the previous one is kind of like high resolution, the deep deep coverage of the tissues, but limited antibodies or limited markers you can measure. But for the RCMS part, if you want to discover something, or if you want to have thousands of proteins, then it is the way you should use. So among these RCMS-based spatial proteomics workflow, one good representative is called deep visual proteomics, developed by proteomics pioneer Matthias Mann from Max Planck in Germany. And in that work, it can achieve the single cell resolution through the AI-guided imaging and to get the laser capture microdissection to collect the areas of interest. And then they use the high resolution and high sensitivity proteomics instrument like the timsTOF or Astra, and then they can do very high throughput and depth analysis. And we can see that for this type of work, it's mainly the laser part which decides the resolution. However, this laser capture microdissection, or we can call it RCM-based workflows, sometimes may face some challenges in achieving single cell or subcellular resolution. Like this study here, you can see that the x-axis is the dissection or laser cutting diameter of the samples, and the y-axis is the copy number of the mitochondrial DNA. And when it's approached to 20 micron, the DNA copy number is approaching to 0. Kind of like the working limit of laser is around 20 micron. So that's kind of beyond or around the single cell. But if you want to go higher resolution, it's impossible. Besides, the capture rate and the sample size has a positive correlation, like you can see here. Because if you cut it, then you have to collect it, then you can analyze it. So by this left diagram, you can see that when it approached to around like 0.002 millimeter square millimeter, actually it's around like 100 cells. It's already as low as 25 to 30% capture rate. That means if you want to collect 100 samples, you will lose around 70 samples. Therefore, if you want to go higher resolution, the laser part or the laser RCM-based method is kind of infeasible. So we think maybe increasing the resolution seems quite critical. But how we can do that if you want to go that below of the resolution? Then the expansion microscopy caught our attention. Actually, this technique is originally used for super resolution imaging developed by Edwin Boyden from MIT in 2015. And we can see here, it's quite similar to the SDS-PAGE gel, but there are several different parts. Like here, we use the sodium acrylate, and like here, provides the ionic groups that may make the gel expand. And the acrylamide forms the structure backbone of the polymer network. And these two are the monomer that may form the polymer. And there are also some cross-linker, like here. They use the methylene-based acrylamide. This one serves as the cross-linker to cross-link this polymer and to stabilize this whole three-dimensional hydrogel. And when you in situ anchor your tissue into the hydrogel, then after you embed it into the hydrogel, and then when it soaks water, it will start to expand due to this sodium acrylamide. And this is a video to show you how it works. Like these brown ones are the molecules of interest. Like this one is the protein or something else like lipid or nucleic acids. And this one in situ anchored because the protein may have this free amino group, and that one can be anchored by an NHS ester and into this polymer hydrogel system. And when this whole system expands, those in situ anchored molecules of interest will also go with them isotropically. And on the right, you can see this is a good example of how a mouse brain slice showing how the tissue expansion actually looks in practice. After you embed the sample into the gel and after several rounds of water exchange, like overnight, it may just like this expand isotropically. Yeah. And this one is also because, as I mentioned in the beginning, in 2015, it's firstly established or developed, and last year it was the 10 years of this amazing method. So last year, Nature has a technology feature to introduce this method, and because it's originally used for the super resolution imaging, and you can see clearly, like on the right, this is the one with the conventional microscope, and together with the expansion microscopy, and the resolution of the performance you can see is quite clear. When it's compared to this one, is like the conventional one without any kind of super resolution technique, then it's quite bad, the resolution. And this one is the normal one, just use a high-end super resolution microscopy. So we can see just with a very conventional microscope and plus these types of expansion, then you can have very good results, even better than the super resolution microscope sometimes. So this is a very cheap way, but can give you very good results. So this is about expansion microscopy's performance imaging. But we think whether this physical magnification could also enhance the sampling resolution in the MS-based spatial proteomics. As I mentioned, for this area, the critical part is the resolution enhancement, but the laser has its limitation. So we think maybe there is a good fit for both. So in 2022, we combined this expansion microscopy technique with ProteomEx as our first generation of expansion proteomics work. We call it ProteomEx. And under ProteomEx, there are mainly three parts. The first part is the tissue expansion. Actually, as I mentioned in the beginning about the principle of this method, and we may know that it is mainly involved in several different types of chemicals, like the monomer from the polymer, and the cross-linker and anchor. So we have validated the combination across hundreds of different types of the hydrogel formulations to identify a proteomics-friendly method, meaning, for that one, it's mainly capture the proteins and has the best protein yield or peptide yield for good mass spec data acquisition. And during that time, we mainly use the manual microdissection method. After you expand the samples, and we can just use a biopsy, clinical biopsy puncture to collect the samples. Like here, we can collect you can use 1 millimeter or 2 millimeter or whatever you want, to use to dissect the samples. It's easy, and you do not need any high-end instrument. And when we collect the samples, and we do the normal in-gel digestion, and then send these peptides for the mass spec for data analysis. Actually, there are quite a few aspects about how we develop this and compare to the other conventional methods like in solution and the other way. But because this part has already been published, you can have a look of this paper. And I will not go to that details because we have quite a few iterations of this method. But I will show you one thing, because during that period of time, we would like to know if we just do this kind of method, can we find something interesting? And we applied the ProteomEx to the 80 mouse models. And here you can see just by punch this 1 millimeter size, we can get around 166 cells. And from this mouse brain on the left, it was the old wild type, and on the right is old AD. And there are different areas of the brain, like the cortex and like the hippocampus and different areas. And we found quite a few molecules, but one of them is the STXBP2. We found it's rarely reported in AD diseases, but we found that it's negatively correlated with the AD. So that means this work has the capability to find the molecules spatially. Yeah. But still, after we publish this paper, we think there are still some limitations in ProteomEx, like the compatibility issue. Because for that part, we mainly do the fresh frozen tissues after a brief fixation. But if you want to do the extracellular matrix-rich tissues, and also FFPE, the formalin-fixed paraffin-embedded samples, is not that easy because of this kind of methylene cross-linking that may hinder this peptide or protein cleavage site accessibility. And another thing is the limited throughput, because for ProteomEx, we are still using this manual method. Then it's long processing time and no automation. So it's quite time consuming. And for this, we may introduce another limitation called the depth and reproducibility challenges, because a lot of manual steps, we may have peptide and sample loss during this whole procedure. It's like 10 to 20% of samples may be lost. And another thing is the limited resolution, as I mentioned previously. It's just can be collected through a puncture like 1 millimeter, then it's just equivalent to around 0.37 nanoliter, which is equivalent to around 160 cells. Apparently, it's not single cell resolution. So there are some limitations. So based on or targeted on, at these limitations, we further developed a new method. We call it filter-aided expansion proteomics, short as FAXP. And for FAXP, it mainly improves the compatibility. Like this part is mainly targeted to these FFPE samples, especially those ones with the extracellular matrix-rich tissues. And we optimized the sectioning, embedding, and the homogenization steps. Especially the homogenization steps, we change the homogenization buffer and use harsh conditions like autoclave. That means high temperature and high pressure. Those things may help to break these methylene cross-linkings. Another thing is we apply the reduction and alkylation onto the whole sample hydrogel composite. Then all the things may help to break down this methylene cross-linkage. And another thing is within this type of tissues, after you have done all these kind of things, it will be far more easier for the enzyme to go inside the hydrogel and to approach the peptide cleavage site, and then to easily to cut the peptides, cut the proteins into the peptides, and then collect them. And another thing is, as I mentioned about the resolution. So here we combined this expansion microscopy method after expand, and then we combine it with the laser capture microdissection. Because the RCM has like around 20 microns limitation. So if you expand the samples, then it will be easier for the whole system to go below to like the single cell or even subcellular resolution, like 10 even to 5 micron is easier. Because think about it, if you do like a linear expansion like 4 to 5 times, if nuclear before expansion is 8 micron, after 4 to 5 times of expansion, it may go to like 30 to 40 micron. So it will kind of like is higher than the limitation of the laser capture. And also it's bigger, then it's easier to collect. And another thing is about the throughput. We developed, firstly, we developed filter-aided. This is how it's named, filter-aided. We developed this small tip. We put the hydrogel into this tip, and then we have this kind of filter-aided in-gel digestion, the whole workflow to help us to decrease the manual steps and increase the peptide collection rates. And also, we built this whole thing into a home-built robotic system, and the whole thing may delineate these manual steps, and we can do like 96 samples per plate, and we can do like multiple plates altogether, and then we send them into higher resolution or sensitivity mass spectrometer, Astra, yeah, which is quite cutting edge mass spectrometer currently, and for better data acquisition and performance. There are some results. Firstly, like we can show that for the FFPE samples of the mouse liver, it can expand isotropically. Like this is around 4 to 5 times linearly. This is like a macro level. And for the for within the cells, how it looks like, because even though we can see like from the tissue level, it may increase, But how about those organelles, within the cells, how it looks like? It's also very important. After we optimize this embedding and hydrogel formulation, we can also make sure this nuclear or the content within the cells also isotropically expand, equivalent to the linear expansion factor as to the macrophage level. And another thing is we can see here, this is the dye staining, and you can see, yeah, after you adding water, the whole area can expand uniformly. And also about the subcellular structure visualization, we have tested the FAXP imaging capability on both mouse liver tissue and hello cells. And we stained nucleus and mitochondria. The green ones are the mitochondria, and the red ones are the nucleus. You can see like for the mouse liver, it's around like 38 to 40 as I mentioned previously. And for the hello cells, it can go over 50 micron. And also the green dots can be stained easily and clearly visible. And the average diameter of the mitochondria after this expansion is around 2 to 3 micron. Yeah. And another thing is about, because normally after expand, it's not only the 2-dimensional, it's 3-dimensional. So how it looks like the 3-dimensional when it comes to the Z-stack, how it looks like. Actually, all this kind of test is done by Zess. Yeah. So we think it's quite promising. We can see here for this 3D imaging, we have imaged Z-stack depth is around 126 micron. And we can see here, the side view shows the intact tissue structure. And you can see it's quite clear, if we go the different Z-stack. And the puncture display, you can see here, shows a typical hourglass shape, which shows it's quite good, even this thick samples after expand. And another thing is, as I mentioned, within the FAXP method, we have a customized automated system with the integrated FAXP workflow. So like you can see here, we have this kind of robotics to help us to collect the samples and to do the whole filter-aided in-gel digestion approach. And for this way, we can decrease the time from like 140 to only around 28 hours. And we can collect like hundreds of samples together. And we can do this, the hundreds of samples together for the mass spec. Yeah. And besides to test the robustness of FAX workflow, we integrate this FAX with laser capture micro dissection. And you can see here is clearly on the left. If you just cut the samples, actually it's the mouse liver's nucleus is around 8 to 10 micron. If you just cut them without expansion, it may look like this. It's totally burned. Yeah, you cannot collect it well. But once you embed the thing into the hydrogel after you expand and then you collect it again, now you can see, you can clearly collect these samples and it's easily to be collected and to do the downstream analysis. And by using this work, we have done this. We have used this ASGR1 specific hepatocytic marker to stain these hepatocytes, and then we isolate independently three different groups. One is nuclear only. Another one is the hepatocytes with one nuclear, and the third one is the hepatocytes with 2 or binuclear. There are totally 3 groups, and then we compare them. You can see that from these tissues by using only by analyzing only the single nuclear, we can detect around 2,700 proteins. And for the cells with either one or two nucleus, we can detect or we can identify over 3,000, close to 3,400 proteins. And compared to the blank, it's significantly different. And within each samples, the reproducibility, you can see that you can see that the correlations are quite high as well, with the values above 0.86 among the nuclei and above 0.92 within the hepatocytes. And the protein expression analysis also revealed that you can see here the nuclear associated terms like the nucleoplasm, splicing, or the other stuff, relevant stuff are more expressed in nuclear. And in contrast, the terms related to the mitochondria, ER, and Golgi apparatus, and the peroxisome are highly enriched in cells, which makes sense. And also, the pathway is the same. Nucleus associated, like the metabolism of RNA or nucleases, are highly activated within nucleus. And also the TCA cycle and the metabolism are highly activated in cells. And all those things prove we can clearly identify and analyze the subcellular proteomics spatially. And as a result of these efforts, the expansion proteomics exemplified by FAXP was just selected as the technology of the month by Trends in Biochemical Sciences. And given the FAXP's progress and its clinical potential, we recently published a standardized protocol in Nature Protocols for wider adoption. And you are very welcome to try this workflow and give us feedback and to make this workflow better. And apart from achieving the subcellular resolution, FAXP has also been applied in several clinical studies by far. And this figure illustrates one example in the intrahepatic cholangiocarcinoma. Here we combined the imaging mass cytometry, spatial proteomics, namely our FAXP method, and single-cell transcriptomics, spatial transcriptomics, and other stuff altogether. And from this integration, we have found the three different clusters of immune cells. One is immune infiltration, the second is immune tolerance, and the third is immune desert. And by analyzing the phenotypes and the cell neighboring and the areas of interest altogether, and we by analyzing all those kind of things together, especially the tumor microenvironment, the different types of tumor microenvironment were characterized. And furthermore, the spatial deep learning models were applied to extract the key spatial phenotypes and link them with the clinical outcomes, like for the personalized diagnosis or the treatment's outcome, something like that. And this is another example. This showing its application in the colorectal cancer. And normally, for the metastatic colorectal cancer, the MCRC patients, there are mainly two subtypes. One is the microsatellite stability, and another one is microsatellite instability. Either of them may face the same intrinsic resistance to the immune therapy. So, however, we still do not know the spatial tumors TME, the microenvironment, and how this environment distinguish this kind of immune responders or immune non-responders under each subtype. So we want to understand this microenvironment and how this contributed to this responder or non-responder within either subtype. So in this study, we have like different cohorts. The first cohort is used for the imaging mass cytometry and the FAXP spatial proteomics. And second one is used for the spatial proteomics and some others from the external cohorts, like the IC RE-seq, the bulk RE-seq, and some like a bulk proteomics, all those things together. And here we can see that we can find from either subtype, we can find for the responder, they are the same, like the C1QC positive residual tissue macrophage and the CD4 positive T cells are highly localized. And at the same time, the fibroblast is done regulate or has less interaction. And at the same time, the MHC2 molecules may contribute this interaction. But for the non-responder, is totally the opposite. So from this, we can see the capability of FAXPs integration with other, multi-omics studies and for this clinical translation and the compatibility with the different types of clinical tissues. And further, we think actually this is the newest work, and this work is done together by me and Shayao together. And this is about we think this is our first time to talk about this publicly. This about what is like because now that we know that for the spatial proteomics, it may face its own challenge as mentioned, but there is a data dilemma. We think, like we can see here, we have one method to achieve host light, but low resolution methods, like last year's cell paper, Plato, or another cell discovery paper, S4P. This both methods use different ways, but they can achieve entire tissue section reconstruction through the AI model together with the mass spec method. But either method only can achieve the resolution above like 25, and some like S4P even go beyond like 300 micron. So it's far more away from the single cell resolution. And another way, as I mentioned in the beginning, the DVP method, they can achieve relatively high resolution, but they are still area of interest based. That means you cannot do the whole section. You have to have the prior knowledge which part you would like to be to collect. So that means it can provide the single cell detail, but only for a small area. So if we want to do this host light high resolution methods, currently there's neither way can achieve that. So that means this leaves us a critical gap. So we think the ideal technology would combine both ways. That means we can do the host light coverage at single cell resolution. And this is the technical bottleneck. What this following, FAXP, we call it FAXP 2.0, is designed to address. Due to the time limit, I will not go to that detail. But here you can see it's just this is the experimental design. We use the consecutive slides, like 2 slides, and we can do like after expansion, we can do the expansion microscopy imaging. That means high resolution imaging as the reference. We can do this or some other ways like codex, whatever, we just need this kind of immunofluorescent images as an input for the reference to get the reconstruction. And these 2 slides, we will do the orthogonal sampling. One is for this, you can see that we collect like M column, M rows here. And here we can collect N number N's columns. That means this is orthogonal sampling. And you can see that for this orthogonal sampling, there will create this kind of patch. And this patch may, because after you expand, we cut around like 50 micron. So if you divide by this expansion factor, it can go to around 10 to 12 micron. It's close to the single cell resolution. And then we do the FAXP sample preparation and to do this high resolution and high sensitive mass spec data analysis. And as input, we have this kind of immune fluorescent images and also this row and column expression matrix. And we developed a deep learning model. We call it HER2-NET. And this is a kind of orthogonal pressure-based CNN model, and it enables the accurate reconstruction of the spatial proteomics maps at this kind of single cell resolution. Because normally we collect or we identify around like thousands of proteins, and so it can reconstruct this whole thousands of proteins of back to this like here back to this spatial at the single cell resolution. Yeah. And there are some validation, like with this robust workflow. And firstly, we start with some well-known liver donation markers to validate the robustness of our work. And we use like GLUL, which is a marker for the central vein area of the liver, mouse liver or the liver donation. And the ECAD as a portal vein area marker. And on this left is the image, the immunofluorescent image we collect. On the right are some well-reported or well-known zoom markers, like the CYP 2C29 or like this one. These are reconstructed by the HER2-NET. And you can see this reconstruction accurately or you can see accurately mapping to this ground truth like CV or PV. And further, we also tested the other predicted CV or PV markers. All these markers are from a Nature Methods paper published by this month, and they prove these things are highly relevant to CV or PV, and we can reconstruct all these 20 markers, and it's relevant or accurately equivalent to this how you call it distribution of the CV or PV zones. And further, you can see here we pick an area, and within the area you can see the each patch is like around the single cell resolution, and then here is like the CV area, and here is the PV area, and we pick this the different you can see that beyond the single proteins, we wanted to ensure our model could capture the complex biological functions. So we within this area we pick randomly pick several patches, and we perform the gene site enrichment analysis across the reconstructed spatial map. And the results on the right showing the normalized enrichment scores for selected gene sites in each spatial beam compared to all other beams. And we observed a clear functional gradient from the central to the central vein to the portal vein. And these findings, like these 6 pathways, are in complete agreement with the established liver biology, proving that our AI model reconstruction is accurate. And further, we applied this HER2-NET to invasive colorectal cancer samples using the multiplexed codex imaging. It's around 30 protein markers as input. And the model predicted the spatial expression. This is a TMA core of the colorectal cancer from human. And we used the HER2-NET to predict over 2,000 proteins. And the first line is the ground truth extract from the codex imaging. And the second and third rows are the HER2-net reconstructed results. But we can see that for those kind of clinical samples, especially very heterogeneous samples, higher numbers of features of the references, like the codex references, may reconstruct a better or more assembly to this ground truth compared to lower numbers of features. Actually, this work has already been pre-printed and it's available here because there are quite a lot of other validations and the methods and also how we apply this to the clinical samples. So if you are interested, you're welcome to check this preprint paper. And the third part is about how we use the spatial proteomics as a foundation for the AI-driven virtual cells. Recently, the Nature featured the spatial proteomics as a cornerstone for building the virtual cells. The article interviewed my lab PI, Dr. Tiannan Guo, and highlighted our work as well. Actually, we can see there are two things. Firstly, I want to share is like Nature think the expansion microscopy-based proteomics work is a very good way to make this MS-based protein measurement easier and more affordable. Yeah, so that means it's easier for better adoption. And another thing is, Dr. Guo think all this kind of data, especially the spatial data and the multi-omics data, they are all helpful to build a fully digital model for the human cells, namely the virtual cells. So it's a quite emerging approach. So also last year, we published a perspective to about show how we vision this kind of AI virtual cells built. There are mainly three pillars, we think. The first pillar, we call it a prior knowledge. That means like the literature, like the database, like there are all the images already published, all those kind of things as the prior knowledge. And the second pillar, we think, is the static architecture. We can do the living image and the like omics data set and the dynamic like perturbation. And the third pillar is the dynamic states. It consists like the natural process, like the physiological processes like disease, and also the perturbations like the drug treatment or like the CRISPR knockout, something like that. And we think robotics may help to accelerate this whole process for the automated data generation. And in the long run, we hope it may become a closed loop active learning system. So how the expansion omics contribute to the AIVC? As I mentioned, the three pillars. We think, so here we think this is our vision actually. This is also a perspective we published last year. We call it the expansion omics from biological samples to the AIVC construction. Firstly, as I mentioned, the current mass spec-based proteomics may do quite a few different types of samples. So does to the expansion proteomics. It can do a variety of biological samples like cells, different tissues, different organs, even to bone, and even different organisms. And we think through the tissue expansion, the resolution, as I mentioned, can be increased up to if we do like a one time expansion, it can do like 3 to 11 times based on the publication studies, around like 27 to 1,000 over 1,000 in volume increase. But Also, there are some other ways to do the iterative expansion. If you do that way, you can even go far, go higher, like 13 to 22 times linearly, and you can go up to 10,000 times of fold in volume. So these things enabling like the super resolution imaging and the spatial multi-omics, and even the molecular architecture, like the protein structure or protein complex analysis. And this framework, when you combine with this AI-driven computation strategy, as we mentioned in the FAXP 2.0 method, when you combine with those kind of model, we can get this whole-slide molecular mapping at the single cell or maybe even subcellular resolution. And in the long run, it definitely can provide the high-resolution static architecture, namely the second pillar within this AVC here. Yeah. And actually, if you want to do dynamics, it's also, I think, it's because it's just like a time series or something else. So we think in the long run, expansion omics may provide the high resolution data set for constructing the AVC. So in conclusion, we developed this expansion proteomics exemplified by FAXP. It combined the tissue expansion with streamlined proteomics workflows to enable the high throughput, reproducible, and high resolution analysis. And we vision it may provide a robust platform for spatial proteomics of this FFPE and some other clinical samples. And it may enable the multi-omics and structure mapping and supporting the construction of the AVs. Actually, we are already, because we have already published this Nature Protocols paper, and we also make some kit as shown on the left. And also on the right, there's a QR code. They are free to have a look. This is a website. We call it FAXP, but within this website, we have already collected all this information regarding the development applications and also other information. If you want to know more details, yeah, welcome to scan it. And in the end, I would like to thank our collaborators and the supporting grants. And I'm happy to take any questions. Thank you for listening. Yeah.
Okay, thank you, Dr. Zheng, for this insightful and inspiring presentation. And we now have some time for questions from the audience. Due to the time constraints, I will choose 3 questions on the chart. So first question is, do you have any idea of the peptide retrieval efficiency after laser microdissection GLP's collection?
Peptide retrieval. Yeah, I mean, you mean peptide retrieval after laser capture microdissection of what?
Peptide retrieval efficiency after laser microdissection GLP's collection.
GOP, what is GOP?
GLPs.
GLP.
Yeah.
I say it because I'm not sure what is GOP. Yeah.
就你有什么提高回收效率的方法在 LC？
Okay. Okay. I think for the peptide because I'm not sure if this question is about the expansion or not, because after you embed the samples into the hydrogel, we expand it, as I mentioned in the beginning, during the introduction of this method. Because after expanded, the peptide, the proteins may also expand. So that means it may be very flexible and it's easier for the enzyme to go inside. So within this approach, it will be relatively easier for the enzyme to digest the peptides. So I think we have already actually compared after expansion with a laser capture, we compared to the conventional, like without this expansion. We can see higher peptide retrieval rates. So we think this is one thing. And another thing is, if you want to increase the peptide, because laser, no matter what, it may cut or it may damage the samples because laser has this energy. So I think if you want to have higher performance or identification, definitely you cannot go to that small size. Maybe for some cutting edge lab, they can do something like that. But even for us, we have tried. If you do very small size of the samples, it's not easy to collect them, and definitely you cannot get enough peptide. Yeah, I'm not sure if I answer it right because I'm not so sure the question. Yeah.
Okay, and also this question, after these questions, have you tried increasing the expansion factor using other expansion techniques to increase subcellular microdissection resolution?
Yeah, that's a good one. Yes. As I mentioned here, sorry. You can see here, actually, there are quite a few different types of approaches tailored for different purposes, like for proteomics, for the other ways. And some other approaches is try to increase the linear expansion factor. Actually, in our lab, now, normally we use is around 4 to 5 or 4 to 8 maximum if you just expand it once. But now, actually, we have some other method, and we can use like iterative expansion. And actually, it's also under our formulation, but we use different strategies and different formulations. And now we can achieve up to like 40 times linear expand. And if you are interested, you can see I think there are like hundreds of different types of formulations that can increase the linear expansion factor. There are also some work you can just do like one shot. You can go like beyond to 20 times linear. So yeah, definitely we try different ways, and yes, it can increase the linear expansion factor up to like 40. Yeah.
Okay, thank you. So the second question is whether the LCM collection is based on the actual cell shape or simply defined as circular regions.
Yeah. We can see, from the FAXP method, the subcellular, as I mentioned, yeah, because we have already visualized or stained the samples through different antibodies, and then you can just see it, and then you can just because you can see all these images are taken from this laser capture microdissection. That means, within this after expansion, because this is after expansion, and then we stain it, and we keep the staining, and then we can just collect no matter what the subcellular areas of the samples as you wish. So you can just count them in the image. Yeah.
Okay, so in addition, how thick are the tissue sections on the slides, and how do you evaluate the level of contamination in the LCM capture samples?
The first part is what?
First is how thick are the tissue sections?
Okay. The thickness, actually, there's a trade-off because, like for the FAXP method, we can do like 10 micron, and for FAX 2.0, we do like between 4 to 10 micron. Actually, there is no limitation. We think we can do like up to like 100 micron. We can also do down to like 1 micron. It doesn't matter. But the trade-off is, for example, if you want to go thicker, definitely you can get higher identification, but the resolution is lower. But if you want to go to higher resolution, like to go there, because the samples are the three-dimensional, it's not only just a plain plate. So if you go like thinner, then you cannot get enough identifications. So there is a trade-off. So no, but normally we do like for the FFPE tissues or the clinical tissues, we do around like 4 to 8 micron. And for the mouse tissues, like brain or liver, 10 is a good balance. And the second part is about what's the second part of the question?
How do you evaluate the level of contamination
Yeah, this is a good one. But yeah, actually we didn't do the the strict evaluation. But as I mentioned here, we can see that if you collect the single nuclear and also you collect the different hepatocytes, you can see clearly the different enrichment within each area. So we think it may not be as easy as contaminated if you do not do the expansion. So we think it may get relatively clean samples. But actually, this is a good point. I think it should be as a quality control if we want to do more applications. Yeah.
Okay, so how do you know proteins are not leaking out of the membrane of expanded cells?
Yeah, this is a good one. As I mentioned in the beginning, you need to anchor the samples. As I'm, oh, you can see here. These brown ones are for example, if you for us for proteomics, we definitely want to capture all the proteins. So firstly, you need to anchor the proteins in situ. How we anchor it? For no matter what types what types of proteins, they may all have this free amino groups, like the lysine, this amino acid. So for those kind of amino acid, it can be anchored through this NHS ester into the hydrogel. That means before we want to collect the samples, all these proteins are fastened tightly within this hydrogel. But if we want to collect the samples, we may increase the pH, and this linker or this anchoring is reversible. Then we can collect all the samples of it, all the peptides of it. So by using this way, the peptide yields is quite good. Actually, as I mentioned, if you are interested, you can have a look in the 2022 Nature Communications paper. And within this paper, you can see that we compared by using our expansion method to the in solution method. And actually, we even get higher peptide yield compared to the conventional method. That means we didn't lose much of the samples. Yeah.
So the last question is an open question. With the increasing dominance of AI and computational approaches in life sciences, what's the outlook for experimentalists in structural biology or biophysics?
Yeah, I think this one you should have me too. Yeah. I think this one Shayao should have me to answer. Yeah, as I mentioned, Shayao is a collaborator, and actually he's mainly doing this AI part of this FAXP 2.0. So we have a deal. If there are some AI relevant questions, I think Shayao can help to answer. Yeah, I think it's.
I think I think the experimentalist maybe because now the web coding is more hot, so so the coding technique for experimentalist is more easy. So you can learn it for yourself, and You can also collaborate with other AI lab or AI group to we can say that the integrated AI multi-AI model, yes.
I think I will add one. I think this is a good questions for a lot of experimentalists because people may think AI may do a lot of stuff and what's the use of our experiments results. But I would like to say, no matter what, data is very important. Yeah. I think the same for this AI or the computational models. There is a saying like a rubbish in, rubbish out, right? Yeah. So data is if you want to have a very good reconstructed results, or you have to have very good quality data. So I think we should find a good fit into this how you call it, this kind of ecosystem. We need to find a good place to provide high quality data, and then by using AI as a tool to help us to get something beyond our data, but not just have no data and you use only use AI to give you all the things. I don't think these things are credible. Yeah. So still, I think experiments are very important, but good quality data is the key for the AI area. Yeah, that's my comment. Thank you. Yeah.
Okay, thank you for, due to the time constraints, we will conclude today's session here. And thank you, Dr. Zhen, for sharing your outstanding work and thoughtful perspectives with us today. And thanks to everyone in the audience for joining us and for your engaging questions.